Welcome to Oncolife interview session. It's my pleasure to introduce our guest today, Dr. Thomas Heineman, Chief Medical Officer of Oncolytics Biotech. You are welcome.
Thank you very much. It's good to be here.
In this session, we will discuss the potential of the oncolytic viruses, their role in cancer treatment, and the latest data from the BRACELET-1 breast cancer study. Dr. Thomas Heineman, could you share some information about your professional journey and the mission of the Oncolytics Biotech?
Sure. So my name is Thomas Heineman. I'm the Chief Medical Officer at Oncolytics Biotech. I've been with the company now about 4 years. My background is as a physician and a medical scientist with a doctorate in virology. I've spent about a decade or so in academia, seeing patients and doing research, and another decade or so in big pharma, especially at GSK, where I led the development of a number of their different vaccines. Over the past 7 or 8 years now, I've been working exclusively in oncology and immuno-oncology, developing new treatments for a variety of different cancers. And here at Oncolytics, our mission is to develop immunotherapeutics for cancer, with our primary asset being the oncolytic virus pelareorep, which has shown substantial promise in a number of different cancers, particularly breast cancer, pancreatic cancer, and some other gastrointestinal malignancies.
Well, the oncolytic viruses is very interesting topic.
Mm-hmm.
They are not only destroy cancer cell, but also modulate the tumor microenvironment to enhance anti-tumor immune responses. Could you provide some information about oncolytic viruses and their role in cancer treatment?
Sure. So the oncolytic viruses are actually very promising class of therapeutic agents, and there's a very broad range of them. These oncolytic viruses vary quite a bit from one another, and they have the attractive properties of being able to naturally infect and in some cases replicate in and kill cancer cells. I think that what has become increasingly apparent over the years, and this is true for pelareorep, which is the oncolytic virus that Oncolytics Biotech is developing, is that oncolytic viruses in most cases function primarily by modulating the immune system, more so even than direct killing of the cancer cells. So, for example, the oncolytic virus that Oncolytics Biotech is developing, pelareorep, is a naturally occurring isolate of a reovirus which selectively infects cancer cells. And what makes it distinct from many oncolytic viruses is, one, it is not genetically modified in any way, so it does not need to have any special handling precautions. And even more importantly, it selectively infects cancer cells. It does not infect normal cells. So when it's delivered to patients, it will target the cancer cells directly, and it can be administered intravenously. So many oncolytic viruses require intratumoral administration because they can be neutralized in the bloodstream if they're administered intravenously. Pelareorep can in fact be administered intravenously because it evades neutralization in the bloodstream. So that allows it to directly target tumor cells wherever they are, whether it's a primary tumor, a metastasis, or some site that may be inaccessible through some other means. And also, not more importantly, but importantly from a patient's perspective and a physician's perspective, it allows pelareorep to be delivered simply, safely, conveniently in a oncology suite, for example, rather than requiring any special handling or administration procedures.
Thank you. And could you elaborate on the mechanism of action of the pelareorep and how it stands out from other oncolytic viruses in terms of the efficacy and safety, particularly in HR positive HER2 metastatic breast cancer?
Yes, so certainly. So pelareorep is actually a very unique agent among the oncolytic viruses. It's a reovirus, and reoviruses have double-stranded RNA genomes, okay? And so when pelareorep infects cancer cells, and as I mentioned, it infects cancer cells selectively, it releases its RNA genome into the cancer cells, which is recognized as foreign by the cancer cells, and stimulates an inflammatory cascade that includes the upregulation of a variety of different chemokines, which then make the tumor visible to the immune system. Okay, so that's one element of it. Simultaneously, while that's happening, pelareorep also upregulates or causes the expansion of tumor-infiltrating lymphocytes, which can then target the tumor. And so the way we believe pelareorep works is that it stimulates these anti-tumor cellular immune responses, and because it's modifying the tumor microenvironment and making it more visible to the immune system, it allows these anti-tumor immune responses that have been stimulated to enter the tumor cells and directly target the tumor itself. So it has a what you might call a dual mechanism of action that makes it a very efficient agent for targeting tumor cells. Now, specifically for the HR-positive, HER2-negative metastatic breast cancer, this is a population in which we've seen particularly strong clinical data demonstrating pelareorep's benefit. And in this particular tumor type, immunotherapies have not been very successful historically, but because of pelareorep's ability to modify the tumor microenvironment, we believe that it is making these tumors more responsive to immune responses. And we see this in the data derived from some of our studies, in which we clearly see in these patients an upregulation of T cells, the expansion of T cells in the blood following the treatment with pelareorep.
Moving to the next question, the BRACELET-1 study showed a median overall survival of at least 32 months in the pelareorep and plus paclitaxel arm. What are the key factors contributing to this significant improvement?
Yeah, so in the BRACELET study, in this study we compared pelareorep plus paclitaxel as the investigational arm to the control arm of paclitaxel, which is just a standard chemotherapy alone. Okay, and in that study, we showed that in the paclitaxel alone arm, that is to say the control arm, the patients had a median progression-free survival of 6.4 months, which is typical for that type of treatment. When we added pelareorep to the paclitaxel, we saw that their median progression-free survival went to 12.1 months, a near doubling of the progression-free survival. And then, as you alluded to, for the overall survival in that study, the paclitaxel alone patients, the control patients, had a median overall survival of 18.2 months, which again is very typical for this population. And for the pelareorep arm, the median overall survival actually was not even reached in the study, because at the end of the study, more than half the patients were still alive. However, if we took the very, very conservative assumption that at their next visit, all the patients would have died, which we certainly hope and expect would not happen, but if we take that assumption, then even with that conservative assumption, the median overall survival would have been over 32 months. So you can see there's a strong progression-free survival and overall survival benefit in the pelareorep arm. And we think that this is happening, again, because of pelareorep's ability to make the tumors visible to the immune system and to stimulate these T cell responses, which we see in these patients. So the T cell responses that are capable of targeting the tumor are stimulated by the treatment with pelareorep, and they are made accessible to the tumor by its inflammatory effects on the tumor microenvironment.
These are really important data you shared. And moving the next question about the BRACELET-1 study included a cohort combining pelareorep with avelumab and an anti-PD-L1 therapy. Could you share any preliminary observation on how the pelareorep modulate the tumor microenvironments and potentially enhance the efficacy of checkpoint inhibitors?
Yes, certainly. So in this study, we did have an arm that included the checkpoint inhibitor avelumab, and in fact, the avelumab did not add any benefit to the treatment in this particular case, and that I think has to do with the nature of the checkpoint inhibitor. Avelumab is a unique checkpoint inhibitor in that it retains binding of the checkpoint inhibitor to what would be called the FC receptor, which actually has a paradoxical effect of eliminating the early immune responses stimulated by the pelareorep itself. Okay, so as a matter of moving forward with this treatment, avelumab is probably not the right combination partner. Okay, and as it turns out, we don't need it anyway because we see these strong effects even without it. But having said that, other checkpoint inhibitors with somewhat different designs, I think, have shown some real promise in combination with pelareorep. And we see that in particular in the gastrointestinal cancers, for example, pancreatic cancer. And I think the reason that we feel that pelareorep can enhance the benefit of checkpoint inhibitors in some of these tumors where checkpoint inhibitors do not typically work is, again, because it stimulates this very strong inflammatory response in the tumor itself, which makes the tumor visible to the immune system. So it allows the immune stimulatory effects of the checkpoint inhibitors to actually have an effect on the tumor. And specifically, it also upregulates the expression of PD-L1 in the tumor, which is a molecule that is very important for the activity of these PD-L1 inhibitors. Okay, and so we think that the pelareorep plus checkpoint inhibitors in general provide a very strong opportunity to further enhance the immune killing of certain types of tumors in which checkpoint inhibitors normally do not work.
Well, beyond the breast cancer, pelareorep has shown promise in the pancreatic cancer. What's your vision for expanding its application to other tumor type, and are there any specific cancer indication that you are focusing on for future studies?
Yeah, sure. So we've done a number of studies of pelareorep-based combination therapies in pancreatic cancer, and we have seen some very promising clinical benefits in these studies. The most recent one is something called the GOBLET study, in which pelareorep was combined with standard of care chemotherapy, in this case gemcitabine and nab-paclitaxel, and the checkpoint inhibitor avelumab. And in that study, we saw a very strong tumor response rate of over 60% in patients treated with the pelareorep-based combination therapy, which is much higher than you would typically expect in this very difficult patient population. And so that study, combined with the earlier studies of pelareorep combinations in pancreatic cancer, have made us very optimistic about the potential of pelareorep to provide a benefit in this patient population, which has proven a very difficult area to develop new therapeutics. So we are planning to move forward with the development of pelareorep-based combination therapy in this population. We've been working with an organization called GCAR, the Global Coalition for Adaptive Research, who is very interested in working with us in the pancreatic cancer space and are very experienced in conducting innovative and strong, very strongly designed studies that can lead to licensure for new therapeutics in different areas. Anyway, we've talked to them and to their scientific experts with whom they consult, and are actively in the process of designing our next study to evaluate pelareorep in pancreatic cancer. So we have a path forward there, and we think that there's a very good likelihood of success based on the prior studies that we've conducted. In addition, also in the GOBLET study, we've developed a relatively small data set so far in anal carcinoma in patients who have failed primary therapy. This is a rare cancer and with very high medical need because these patients have very few treatment options and really no good treatment options. And we've seen some very compelling data in a relatively small so far population, but the data we've seen have shown response rates of something in the neighborhood of 3 times what you would expect in this population. So we have expanded that arm of the GOBLET study. We're enrolling additional patients, into that study. And if that efficacy signal remains after evaluation of a larger population of patients, then we would like to move forward with the further development with an eye towards licensure of pelareorep combination therapy in the anal carcinoma population as well.
And what about the long-term safety and tolerability finding from your study involving the pelareorep, have there been any unexpected adverse event or an acute safety signal from the clinicians that
Yeah, thank you. No, I think that I should have mentioned this earlier, but that is one way in which pelareorep is distinguished from other oncolytic viruses that are under development. And that is that pelareorep has actually been in development for a while now and has been administered to well over 1,100 patients across different cancers. Okay, so we have a very large body of data from a wide variety of different cancers that pertain to pelareorep safety. And so we have a very strong understanding of its safety profile. What we see with pelareorep is very typically patients will, not everyone, but a proportion of patients will experience what might be termed as flu-like symptoms. So patients may have transient fevers, chills, muscle aches, things like that. These are usually low grade, typically transient, and typically do not lead to any kind of change in the treatment. Patients can be pre-medicated with anti-inflammatory agents such as Tylenol or Motrin or things like that to help minimize these effects. And so even though we do see some typically minor adverse events related to pelareorep, they are usually not limiting in our ability to use it. And so in these sort of populations where we're addressing cancers that are very difficult to treat with very limited treatment options currently, we think that that pelareorep safety profile is actually very much in line with what would be acceptable for this type of patient population. Mm-hmm.
Well, we are moving to the last question. As you look at ahead, what are the key milestones and the research goal for the Oncolytics Biotech over the next 5 years? How do you envision pelareorep changing the standard of the care for cancer patients?
Yes, so over the next, several years, we expect to develop pelareorep primarily in breast cancer, pancreatic cancer, and if the data continue to look positive in anal carcinoma, there are other areas that we will likely look at over time. I think in the more immediate future, we're looking to we have identified a very clear pathway towards a potential accelerated approval in breast cancer through a large phase 2 study, which we hope to begin enrolling in the first half of next year. And once it starts enrollment, we hope to have a readout on that study and give or take 18 months or so after that point in time. And as I mentioned, we have a very clear pathway defined in pancreatic cancer through our work with GCAR, and we are hoping to move that study forward on a similar time frame. Now, we do need to have discussions with the FDA to make sure that we have addressed any questions or concerns that they may have regarding these programs. But our prior discussions with the FDA have led us to believe that we're on the right path in both of these indications. And so we feel that in the not too distant future, we'll have two very clear development routes in two very different cancers, but cancers in which pelareorep has shown a great deal of promise.
Thank you very much for all this important information you share with us. I don't know, do you have something else to underline in this interview?
Well, no, I mean, thank you very much for the opportunity to speak with you. I think that the only thing I would mention is that pelareorep is really unique among the oncolytic viruses, not only in the type of virus it is, its mechanism of action, but also just the sheer amount of clinical data, both efficacy and safety, that we've generated. And so with the very strong breast cancer data, which I discussed earlier, I think that we have a really, really solid pathway towards late stage development, and we're really looking forward to seeing where this takes us next.
Thank you again. We will be happy to follow your study and to have exciting news from the new data. Thank you very much again for joining us.
Thank you.